Good afternoon, everyone. Welcome to this EBBS and VNA session, Bridging Animals and Human Studies in Episodic and Spatial Memories. My name is Dorothy, Professor Alex Easton here. We are the co-chairs of this session. This session addressed a critical challenge in neuroscience, the translation of findings from animal models to human clinician applications, and also preclinical. So we hope this symposium will spark meaningful conversations and encourage new collaborations that help bridge the gap between basic research and preclinical and clinical practice.
That's great. Thanks, Dorothy. And I'm just going to say a couple of little words before we get started about the European Brain and Behaviour Society, which I think if you're in this room is probably of interest to you as well, and absolutely not promote their meeting at this meeting. But this is the third BNA Festival of Science, Neuroscience that we've partnered with. We've been a partnering organisation, and each time we've managed to have a symposium which I think has been a really fascinating one, bridging all sorts of topics around behaviour, and behaviour is something absolutely keen to the European Brain and Behaviour Society, so it's great to be able to do this once again. For those of you who want to be in the south of France at the end of June, absolutely do join us, and if you don't, membership is cheap and very inclusive, and we're a great little friendly society, so do absolutely find out about us, get engaged and join in. So with that, we'll head into the talks for the afternoon, and we're going to start with Gareth Barker from Bristol. It's going to be a long afternoon, I can feel it already. Gareth Barker from Bristol, who's going to talk about mapping engrams, networks of associative recognition memory.
Hi everyone, can you hear me okay? Great. So thank you to Alex and Dorothy for inviting and organising this symposium. So yeah, I'm going to talk to you about our work on engram networks of associative recognition memory. So associative recognition memory, so what we mean by this is our ability to form connections between items and the locations we experience them in. So, for example, we all sort of have a living room and we all know pretty much where everything is, so that if we ever walk in and someone's moved everything around, we'll quickly recognise that and sort of notice what's changed, and we'll form a new representation of the room. Now, this sort of memory is part of everyday memory. It's sort of fundamental for our ability to function and get round. And one of the interesting things about it is that you see deficits in this type of memory early in Alzheimer's disease. So we've been studying this in rodents for quite a number of years, and other labs have as well. And we know that we've identified a number of brain areas and networks, interactions that are critical for this type of memory. So here's our sort of the brain areas we're most interested in. Others are important as well. So these include the hippocampus, medial prefrontal cortex, lateral entorhinal cortex, and the mediodorsal and nucleus reunions in the thalamus. What I'm actually going to talk today about predominantly is our work recently on the lateral entorhinal cortex, but I'll try and connect it back to the rest of this if I have time. So the work we've done to identify this network has predominantly been focused on manipulating entire brain areas or entire connections, so we affect all the cells. But what we've been trying to do more recently, and what a lot of people have been doing, is trying to use engram models to actually study specific cell populations, to try and target the cells that are activated when an animal has been learning anything. So if you're not familiar with the idea of engrams in memory, that's the idea that was proposed by Sermon back at the beginning of the 20th century, the idea that we have a sparse population of neurons that are activated during memory encoding. That information is consolidated, and then when you retrieve the information, specifically you get reactivation of the neurons that were activated during memory encoding. So over the last decade or so, a number of models in mice have come along that allow us to actually label these cells, or try to label some cells that are active in a specific time window, And the one we've been using is this TRAP2 model here. So this is a mouse mutant line that contains Cre recombinase under the control of a FOS promoter. So the idea is that we will get production of Cre recombinase when the cell is activated, so when you get FOS production. And what we do is we cross that, in this case, with a mouse line that expresses TD tomato in a Cre-dependent manner. And so the case is that when we get activity, we get production of Cre recombinase, but in a normal condition, that won't be able to access the nucleus, so it can't drive expression of our transgene. But when we give an injection of 4-hydroxytamoxifen, that binds the Cre recombinase and will then allow it to enter the nucleus, where it can remove the stop codon on our protein of interest, and we get expression of, in this case, TD tomato, so any Cre-dependent gene that is present in the animal we can control. So in this example, so the cells that will be activated that have had FOS activation after we've given the 4-hydroxytamoxifen injection will express TD tomatoes, and we will putatively view them as our engram cells. So we can then take these engram cells and we can do a number of things with them. For example, so these will give us our cells that are activated during memory encoding. We can combine that with, for example, FOS staining after memory retrieval to look at how these are reactivated during memory retrieval, but also we can do things like taking slices from these brains and then recording from cells and comparing the properties of non-engram and engram cells as well. So what we were trying to do in this study, we had sort of three main aims. Firstly, we were interested in seeing if are LEC neurons activated and reactivated during associative recognition memory formation. Are these LEC engram cells actually essential for memory recall? And finally, what we wanted to do was look at whether or not we have any cellular or synaptic changes that occur that we can identify in these LEC engram cells. So the way we looked at the first aim was we took our TRAP2 mice and crossed them with a TD tomato reporter line, and we ran them, so we'd split them into two groups. We habituated them for 6 days to the arena to try and minimize any non-specific engram cell activation, and then we split them into two groups. So our control condition, the animals were simply placed into, this is a 50 centimeter square arena. They were just simply placed into that for 10 minutes and allowed to explore, taken out for 5 minutes, placed back, and then given an injection of 4-hydroxytamoxifen to label the engram cells. So our behavioral condition, so in the mice, we can't put them in the living room. We have to, what we do is we present them with 4 different objects for this object in place task. And what we expect them to do is they'll associate each object with a location. So they're allowed to freely explore those for 10 minutes. And then after a 5-minute delay, we place them back in, but now we switch the location of 2 of the objects so we could get a memory readout at a short delay. And then we labeled the engram cells there. And then for both groups, we then waited 7 days to allow our TD TOM expression to reach sort of plateau levels. And then we returned them either in the familiar arena group back to the arena and then 90 minutes perfused them so that we could stain for FOS. Or in our object in place group, we now switch the location of the objects that had remained stationary in this first test phase so that they've now got these. So they should preferentially explore, if they remember this location, they'll preferentially explore the objects that are moved here. So yeah, and then we stained for FOS and counted the number of trap cells. So the TD TOM cells will represent cells that are activated during this first phase, and then our FOS positive cells will be represented by what happened on day 7. So just to say, we did analyze the data from the performance of the animals here. So this is a discrimination ratio, which is taking the time spent exploring the moved objects, subtracting the time spent exploring the unmoved objects, and then dividing it by the total object exploration. So 0 basically means the animals can't remember, and anything positive, so these are all significantly above 0, so these tell us that the animals as a cohort did have significant memory. So you can see this is the 5-minute delay. The memory does last through to 7 days, the 7-day delay, and we also calculated this combined discrimination ratio from these two as well, which I'll come back to in a minute. We also wanted to make sure that there wasn't any gross differences in activity level between the conditions as well. So we also analysed the distance travelled by the animals and the time spent in the centre. So this is just the distance covered, and it wasn't significantly different between the two conditions. So any differences we see can't be just ascribed to differences in activity in the arena during these phases. So we counted the whole of the LEC, and interestingly what we found is that for our TD-TOM positive cells, we actually see no significant difference between the animals exploring a familiar arena and the animals doing the object in place task. But we do see a significant difference in the FOS. So we don't have a difference in the encoding, but when we're doing the retrieval, our objects in place are showing a significantly higher level of FOS positive cells. And then we looked at the overlap between the TD-TOM positive cells and the FOS positive cells to identify how many of these cells are being reactivated, i.e. we expect, if it's an engram, the cells to be reactivated here and here in both phases. And what we see is that the percent reactivated is significantly higher in the object in place task than it is in the familiar arena, so showing that this is driving reactivation of our engram cells in lateral entorhinal cortex. We were interested, does this reactivation have any sort of connection to the behavior or not? Is there any connection here? So we just did a correlation, and so that I only had one value, I used this combined value, and what we saw is that there is a positive, does seem to be a positive relationship between the percent of reactivated TD-TOM cells and the discrimination ratio, although it's not quite significant, which is such is life. So we can see that the engram cells in lateral entorhinal cortex are reactivated at a higher rate in the object in place task, and there seems to be a positive relationship between engram cell reactivation and memory performance. So we wanted to look in this a bit more depth, as LEC is quite a large structure and it's been subdivided into a number of distinct areas in the mouse along its dorsal to ventral extent. So it's got dorsal lateral entorhinal cortex next to the perirhinal, then dorsal intermediate, and then ventral intermediate down the bottom. So we did the same analysis, but just subdividing LEC now into these three distinct areas. And what we see for all three of these areas is that they are all significantly reactivated by the object in place task. Compared to the familiar arena. But then we looked also at the correlation between the discrimination and the reactivation. And what we found now is that in the dorsal lateral entorhinal cortex there's a very strong correlation between the percent of reactivation and the discrimination ratio. So we've now got a significant correlation here. But as we go from the most dorsal part of the LEC as we move through to the dorsal intermediate, there is still a positive relationship here, but it's now no longer significant. And when we go down to ventral intermediate, there's actually no relationship here between reactivation and discrimination ratio. So it seems that there's a very strong linking between the reactivation of the engram cells in the dorsal lateral entorhinal cortex and performance in this object in place task. So we've identified that the LEC is reactivated, and there seems to be a strong correlation between dorsal lateral entorhinal cortex activation and memory performance. So we next want to ask, well, are these engram cells critical for the memory performance? So we now took our trap line and we crossed it with a mouse line that expressed the inhibitory dread HM4DI in a Cree-dependent manner. So now our engram cells will be labelled with the inhibitory dread so that we can switch them off during memory retrieval. So we implanted cannulas into the LEC, and these were predominantly targeting the dorsal lateral. It's probably likely that some of the CNO will have also got into dorsal intermediate, but we were mostly targeting that area where we'd seen the strongest correlation. So we ran these animals through the task. So we, again, gave them a sample phase and then a test phase after 5 minutes. We then labelled the engram cells. In this case, we waited 48 hours and then infused vehicle or CNO 15 minutes before the test phase. So if the engram cells are critical for memory performance, the animals will be impaired just at this test phase, where we have got an indication that they have learnt from the first test phase. So what we saw in these experiments is that in the first test phase, both groups were able to discriminate, so they did form a memory, but when we infuse CNO into the lateral entorhinal cortex, these animals are completely impaired, so they're showing no difference in exploration between these two, so they have no memory of where they've previously seen these objects, but our vehicle group have robust memory. So that tells us that we do have that these engram cells in LEC are critical for memory performance. So we have activation in LEC, and we know that these are critical for memory performance. So the next set of tasks, experiments we want to do is look at can we see any differences in the neural excitability of these cells. So these are experiments done by Dr. Paul Banks in our group. So he's a slice electrophysiologist predominantly, so he took animals that had been run through this that had objects in place, engrams labeled, and then 48 hours he re-exposed them again to a novel object location, and then 10 minutes after that he sliced the brain and took slices, and he made patched onto cells, and we decided to target layer 5 pyramidal cells in LEC and layer 2 fan cells. The reason for that was that when we did some histology analysis, we found that these were the two groups of cells which had the highest level of incorporation into the engram. So we found the most engram cells there, so it gave us the biggest population to target. So we patched onto pairs of these cells, and he then popped depolarizing steps into these cells. I will say I'm not an electrophysiologist, so if I get this wrong, I do apologize. So in our TD-TOM positive cells, in layer 5 pyramidal cells, when you put a current injection in to make the cell spike, basically the TD-TOM positive cells fire a lot more than the TD-TOM minus, and this is a significant difference here. Interestingly, also the action potentials in these are slightly shorter, so it's slightly lower peak here on those as well. So these cells that have been in layer 5 are much more excitable than the TD-TOM negative cells. Interestingly, when we looked at the layer 2 fan cells, we don't see this. So the TD-TOM positive cells in the layer 2 do not fire more when he put the depolarizing step in. So these are not more excitable, so these aren't being changed by this exposure to the object. So these engram cells haven't got any difference between engram and non-engram cells in layer 2. And the action potential is the same as well. So it looks like associative recognition memory retrieval is selectively increasing the excitability in these layer 5 pyramidal cells. Now, Paul was really interested in terms of what was driving this change in excitability, specifically how much it was dependent upon what happened just before he sliced the animals. So what he did is he reran the experiment, but now in the test phase, he didn't move. In the test phase at 48 hours, he didn't move any of the objects from where they'd last been encountered. So the animals now aren't updating their representation. They're not incorporating any new information into their engram. And interestingly, what we find now is that then we now don't see a difference in the TD-TOM positive and the TD-TOM minus cells. So there's no difference in the excitability of these engram cells. So it looks like something that happened in this retrieval phase is critical for the increasing excitability in these cells. So what we've thought is, well, maybe it's just novelty per se that's driving this rather than something specifically related to the objects. So what Paul did is that he now introduced in the final test phase 4 completely novel objects. So these are just to see if this increase in excitability is purely due to not seeing something novel. And what we see is that these engram cells don't fire anymore here either, although interestingly the action potential was slightly altered in these cells. It was still significantly shorter. So this tells us that it's the encoding of this new object-place association in the test phase that is driving the change in excitability in these LEC engram cells. So overall, what we've seen in these experiments is that the lateral entorhinal cortex is reactivated by object-in-place memory retrieval, and there seems to be a strong correlation between reactivation in the dorsal lateral entorhinal cortex and task performance. We've shown that these LEC engram cells are essential for associative recognition memory retrieval, and we've identified a change in firing selectively in layer 5 LEC engram cells following associative recognition memory retrieval, and this seems to be driven by the encoding of the new objects location pairs. So there. So what I will say is that we did measure similar experiments across some other brain areas. So we do see reactivation in other brain areas, so medial prefrontal cortex, perirhinal cortex, but this does not correlate with the animal's behavior. So we saw no significant correlations between their reactivation in these areas and performance. And also when Paul recorded from cell populations in theirs. He also found no difference in excitability in these areas, so these changes seem to be very closely associated with lateral entorhinal cortex. So it looks like sort of layer 5 pyramidal cells in LEC seem to be playing a critical role in incorporating new information into existing representations. We think that's quite interesting because a lot of the focus on LEC tends to be its superficial layers which project into the hippocampus, whereas actually layer 5 sits in a very interesting place as it receives the outputs from the hippocampus, and we think a lot of other brain areas, such as medial prefrontal cortex, project into those deeper layers, and it also projects to a lot of non-hippocampal areas as well, so it's well placed to kind of organize a wider memory network. So what we're currently trying to do is look at the anatomical connections of the engram cells in the LECs, particularly focusing on hippocampal and medial prefrontal cortex, with either inputs or outputs. So that's it. So I'd just like to thank everyone in Warburton and Bashir groups, particularly Clea and Zaf, who sort of keep us on the straight and narrow, and particularly Paul Banks, Claire Booth, and Lisa Kinaveni, who were sort of all contributed as much as I did to this project as well. So thank you for listening.
Thank you so much, Gareth. Any questions from the audience?
It was really wonderful. I'm a neuroradiologist based in Coventry. So my question is, what would be the human correlate to engram? And would you think, are they based in some layer of hippocampal, like layer 5? Is there any human correlate to that, if you have to guess?
Well, I think we'd be reasonably confident that, well, I think we'd hypothesize that engram cells do exist in humans. We think there's a lot of overlap between how we form memories and how certainly rodents and mice form memories. Where they are, I mean, again, it's sort of with this, we focused on one little bit and we've got one little bit of the story. There's lots of other parts going on, and there's definitely something interesting happening in the hippocampus. We don't get a lot of engram cells in hippocampus for certain molecular biology reasons, and FOS being a particular promoter, but certainly there's a lot of stuff going on there in terms of how the engrams are formed. We've got one bit, but there's lots of bits that I suspect will be critical for that.
Thank you.
Hi, Gareth. That's very interesting. Thanks. I just wondered, do you know how long the excitability changes last? It's correlated with forgetting.
Good question. I just trying to think. Paul, yeah, so we did do some recordings. We don't have a very tight timeline, but Paul definitely did some recordings, I think 24 hours after re-exposure. And I think there weren't any changes in excitability by that point, so they don't last. So they seem to be quite short-lived, although the memory lasts longer. So I think that's why we think there's something very much being driven by that reactivation and re-encoding of the updating of the representation of the objects rather than necessarily representing that. I mean, it's one particular property of the cells we've measured. There's lots of other things we could have measured, but just time and resources limit what you can look at.
Thanks, Gareth. Brilliant talk. I'm super excited by the engram work. The fact that you need updating is really interesting. So do you think this is a reconsolidation phenomenon that you're tapping into?
Well, no, it's something we talk about a lot because, well, it depends how you think about reconsolidation. Because if you think about reconsolidation as just being re-exposed to the environment, when we just expose the animals to the objects in place without moving any of the objects, you could argue that is reconsolidation. Maybe, I know there's a lot of debate about whether or not you need new learning, so potentially that might be, but we don't see any changes there. So it certainly could be a type of reconsolidation, but it's sort of more updating, I think, more than how you fit reconsolidation and updating together is obviously, different people have different opinions on that.
Thank you. Yeah. Cool. Yeah. Do you want to? Sorry.
Sorry. Yeah.
Yeah. All right. Rand.
So thanks for your talk. My question is, so when you label your engram cells initially, you're exposing your animals to, well, essentially two combinations of the same objects. And then, as you confirm, you test that they're not necessarily novelty related because you change, I presume, the identity of the objects. Do you think you would, that it's possible just because you're using different objects, that's the result you obtain? So my question is, if you use the same object, but say, for example, in a different context, and that is because I'm mainly drawing on a model that presumes that LEC, well, deals with the identity of objects,
Yeah.
while the relationship between MEC and LEC deals with the, well, position too.
That's an interesting question. I'm not entirely sure what would happen if we changed the context. We know LEC, there's a lot of evidence that LEC is critical for associating objects with context. So I would suspect that if we change the context, I would predict probably that we have a different engram that would be encoding those objects. So we might see some disconnection between the engram there and the engram from your context that they've been learnt in, but I'm not sure anyone's ever looked at context changes with objects yet. So.
Just in terms of the engram network that you mentioned at the end, there was just a paper that showed, specifically in the context of dementia, that tau seems to disrupt CA1 bursting activity. And I'm wondering what you think the relationship with that to engrams in layer 5 might be.
Ooh.
And if that's worth exploring, maybe.
Yeah, no. I mean, obviously, as I said, that could, if it's CA1 cells that are projecting to LEC, that could disrupt the LEC engram. It could also do it indirectly because they could be projecting to prefrontal cortex. So it'll be interesting. I think there's some stuff that came out of the Tonegawa lab a while ago showing that actually engrams get bigger in Alzheimer's disease rather than smaller, as the idea is that engrams need to be the right size rather than necessarily too small and too big or equally negative. So it could be that it's actually, if you just get general dysregulation, you'll see disruption of the engram network that then disrupts memory. It may not even be that it's completely gone, it's just sort of slightly less well represented.
Great, thank you so much, Gareth. It's my honor to introduce the next speaker, Dr. Aidan Honor from University of York, and he's going to talk about some latest human memory experiments.
Okay, thanks everyone for coming, and thanks again to the organisers. I want to talk about human stuff now, so no rats, I'm afraid, or mice. And I want to talk about how the human brain possibly supports episodic memory, both sort of immediately after you've encoded an event, but also possibly over time as a function of systems consolidation. First, I just want to thank people in my lab, past and present, and funders, but in particular for this talk, I want to thank Bardo Jørgensen, who started some of the fMRI work that I'll talk about at the end of the talk, and Jen Ashton, who also helped with that project. Okay, so I'm interested in episodic memory and critically sort of how the brain allows for the recollective experience, so how we're able to sort of re-immerse ourselves in a previous life event. And here I sort of have a toy diagram of a sort of prototypical event that we might experience in the real world. So I might go to my favorite coffee shop, I might meet a friend of mine, who in this case has glasses, and they might give me a particular object. And the idea here that sort of really dates back to David Marr is that these individual elements of this prototypical event are represented out in the neocortex in distinct regions of the brain. Note, this isn't anatomically correct in any way at this point. So then there's plausibly needs to be some binding mechanism here. We need to bring these elements together, and the hippocampus is thought to be critical for that, right? So the hippocampus acts as a convergence zone, gets lots of multimodal input from different regions. So plausibly, we can bind this information together into what we might refer to as an event engram. So let's say now, after a day or two, I go back to that same coffee shop, but now the person and the object aren't there. So I want to retrieve that. I want to recollect what I did last time I was in that coffee shop. So the idea here is that the representation of the neocortex for that location will be active because I'm in that coffee shop. And that's going to lead to what's called a partial input into the hippocampus. So partial in this case because the event engram consists of 3 elements, but we only sort of have 1 element here. So it's 1/3 of the pattern that we want to retrieve. Now, because of the way the hippocampus is wired up, and particularly the recurrency in CA3, that's thought to lead to what's called a pattern completion process. So the idea that we can retrieve the other elements of that event. But critically, there's a following step here. So what's thought to happen is that we get a sort of rebroadcasting out of that information out into the neocortex. That's called neocortical reinstatement. And it's that neocortical reinstatement that's thought to sort of allow for that recollective experience where I'm able to remember who I was with, what object they gave me. So let's focus a bit more on that neocortical reinstatement and see how see what might support it over the long term. So this is what's thought to happen sort of in the short term, where the hippocampus plays a role in binding this information together, because there aren't sort of long-term connections between these neocortical representations. But systems consolidation accounts suggest that actually the hippocampus might play a sort of time-limited role. So initially, we need the hippocampus to be able to communicate and reactivate these neocortical representations. But over time, we might build up more direct connections, or there might sort of a new sort of hub might exist outside of the hippocampus that allows for this reinstatement effect without input from the hippocampus. Okay, so this is what's referred to as systems consolidation. So some people refer to this as a sort of transfer of information out from the hippocampus to the neocortex. I don't think that's the correct way of thinking about it. It's about it's about the mechanism that allows for that re-expression of that distributed neocortical representation. So either that process is driven by the hippocampus, or it's driven by another mechanism. I'm just going to call that a non-hippocampal mechanism, but that might be via long-range connections. Okay, so how are we going to look at this in humans? So the first thing I'm going to do is just do some memory tests where I get people to learn these prototypical events. So it's no surprise that I gave you that example earlier, because now they're going to learn triplets of locations, people, and objects, okay? So in this behavioral experiment, which I did a long time ago now with Neil Burgess, we were just getting people to learn these associations. So they have to learn that Barack Obama was in the kitchen with a hammer. They get 6 seconds to think about these things interacting in a meaningful way. So they might imagine Barack Obama kind of hitting a kitchen surface with a hammer, whatever. They then learn David Beckham off his wallet, et cetera, et cetera. So they learn a whole series of these. And then we're going to test their memory for these triplets later on. And the way we're going to do that is cue one of these elements. So in this case, I'm cueing with the person, Barack Obama. And they have to remember that Barack Obama was in the kitchen. And these 5 foils here are locations from other events, okay? Now, critically, for any given triplet, I can cue with the person, get you to retrieve the location. I could cue with a location, get you to retrieve the person. I could cue with a person, get you to retrieve the object. So for any triplet, there are 6 retrieval trials. And what we're critically interested in this behavioral experiment is just, well, if we're forming this relatively coherent representation, then maybe the retrieval trials within a particular event should be related to each other, should be statistically related. In other words, if I cue you with Obama and you retrieve kitchen successfully, you should be more likely to retrieve the object successfully for that given event, okay? So it's basically, if we've got 6 retrieval trials, at the extreme, they should be all correct or all incorrect. I'm not going to go into the details of how we measure this, but basically, I've got this measure of dependency here. This is the dependency in the data, and this is what we need to compare it to, which is basically if there's no statistical dependency at all. So if we see this bar greater than this bar, you can ignore the right-hand one for now, then there's evidence that there's a statistical relationship, that these triplets are acting sort of relatively uniformly in manner. Okay, so now what we're going to do is, rather than present these 3 items on the screen at the same time, we're going to separate out the encoding of these triplets into 3 separate encoding trials. Okay, so on one encoding trial, the participant's going to learn the location person. On a second one, they're going to learn the location object. And on a third one, they're going to learn the person-object trial. So now all the encoding trials for these pairwise associations are independent, and all the retrieval trials are independent. But interestingly, we still show this pattern of dependency, okay? So in other words, what I'm not claiming here is we're creating sort of an event n-gram exactly the same as if we had seen those 3 on the screen at the same time, or if we'd experienced that event, but we're forming some kind of associative structure that behavioral at least seems to perform similarly to if you had actually seen those things on the screen at the same time, okay? So now what we can do, now we've separated out the encoding of these pairwise associations, we can kind of mess about with this associative structure and see if we can turn this dependency sort of on or off. So what we're going to do now, so I call this a closed loop condition because we've kind of got this tight-knit associative structure, this little closed loop. And I'm calling this the open loop condition. So here they learn a location-person association and then a location-object association, but critically they don't learn that final association, okay? So there's still 3 elements here. They're still connected to each other, but it's less tightly bound. And critically, we don't see evidence for dependency here. So now this is quite nice because we've got a condition where we sort of show this coherency, where they seem to be retrieving all of these elements together at the same time, versus quite a high-level control condition where we don't see evidence of dependency, but they're still encoding these triplets, sorry, these pairwise associations. They're still retrieving them fine. It's just we're switching this dependency measure on or off. Okay, so let's take this into fMRI now and see if we can find evidence for this sort of neocortical reinstatement and whether it's being driven by a hippocampal process. So the first thing I'm going to do, so participants sit in the scanner, they do the encoding, they do the retrieval. I'm just going to focus on the retrieval data for today. The first thing we can do is just say, okay, well, where in the brain do we see a bigger bulge response if people are being cued by or they're retrieving a location versus trials where location is irrelevant, okay? So I'll call those non-targets because it might be that you're being cued by the person, you're retrieving an object or an animal in this case. The location is irrelevant on that trial. It's not on the screen, you don't have to retrieve it, so that's a non-target, okay? So we see parahippocampal gyrus for when you're either being cued or retrieving a location versus when it's a non-target. We see midline structures for famous people like the medial prefrontal cortex and medial parietal. And we see lateral occipital and lateral parietal regions for objects and animals. Okay? So this is just sort of confirming what we already know. These elements are sort of represented in distinct neocortical regions. Okay? What we're interested in, though, is whether this stuff is reinstated. And critically, what we think is if in that closed-loop condition, if it's acting in a sort of reasonably coherent manner, that we should see reinstatement of that non-target information. So in other words, if I cue with a person and you're retrieving an object, we should also see activity in that region that's coding for the location, because you're retrieving the whole event. And that should be specific to the closed loop relative to the open loop condition. That's why we've got that high-level control condition. So that's what I'm plotting here. So here I'm plotting the percent signal change or bulge response for the closed loop relative to the open loop condition. So up is greater activity in the closed loop condition. And what you can see is we don't see any difference in the region of the brain that's coding for the cue or the target. That shows that these two conditions are quite nicely connected in terms of, or equated in terms of the sort of memory. But in the non-target region, the region of the brain that's the non-target, we see a bigger bulge response for that closed versus open loop condition. Okay? So consistent with this idea that in that closed loop condition, we're retrieving that non-target element and it's getting reinstated out in the neocortex. So presumably we think that's being driven by a hippocampal pattern completion process. So what we can do now is take this non-target reinstatement for each individual and correlate that with hippocampal activity. And what we see is a nice significant positive correlation. So essentially the amount of activity in the hippocampus for that closed relative to open loop condition is positively correlated with the amount of that non-target reinstatement. So this is just a correlation, obviously, but it's consistent with this idea that the hippocampus is pattern completing and then driving that neocortical reinstatement. So this is all in a single fMRI session. So they go into the scanner, they learn these events, we then test them sort of 5 minutes later. So this is sort of an immediate condition, effectively. So we think the hippocampus should be critical here. The key question I want to sort of focus on today, though, is what happens when we do this after 24 hours. So this is the work by Bardot Jørgensen and Jen Ashton that we published recently. So here what we're doing is people come in on day one, they learn a series of open and closed loops, they then go home, they sleep, they come back in the next day, we teach them a whole another series of closed and open loops, then we put them in the scanner and we scan during retrieval. So now we've got an immediate condition, which is essentially a replication of the study I just showed you, and a delayed condition where they've at least had 24 hours and a night of sleep. And we might think that if the hippocampus plays a time-limited role, fair enough, like systems consolidation won't have finished by then, but it might have at least started and we might see some effects. Okay? There's plenty of evidence that the hippocampus might play that time-limited a role from other studies. So the first thing we can do, again, is just to say, well, where in the brain do we see activity when you're being cued or retrieving locations, people and objects, and we see exactly the same regions as the previous study, so just a nice straight replication. The next thing we want to look at, so this is from the previous study I just showed you, showing that non-target reinstatement for the closed relative to the open loop condition. So what happens here? Here we've got a no delay condition, so this is basically a replication of this effect. This is the non-target condition here, and again we see greater activity in this non-target region for the closed relative to the open loop. We also see a difference in the cue for the no delay condition, I'm not sure why that is, but we replicate that non-target reinstatement. And critically, we see the same effect at delay. So after 24 hours, we're still seeing this reinstatement of that non-target information. We're still retrieving these events relatively holistically. The critical question is whether the hippocampus is still driving that effect. So this is the previous study that I showed you with that positive correlation between hippocampal activity and non-target reinstatement. This is the no delay condition. So this is a direct replication of this effect here. And you can see a really nice, strong positive correlation here. So we do replicate the previous effects. This is the delay condition after 24 hours. And critically, we still see a positive correlation. Okay? So the hippocampal activity after a 24-hour delay is still correlating with the amount of non-target reinstatement across individuals, suggesting the hippocampus isn't completely not contributing. But what I want to focus on now is this, this shift in intercept here. So here you can see that this goes straight through the 0 mark, okay? So what that shows is if we don't really have any evidence of hippocampal activity, we don't really have any evidence of non-target reinstatement. This positive intercept here, though, suggests that in some situations we have evidence of non-target reinstatement, we have evidence of neocortical reinstatement in the absence of any clear evidence for a sort of hippocampal effect. So what that shows is that this might be a marker of a non-hippocampal contribution that starts to emerge after 24 hours that might also add on to the sort of hippocampal contribution we're seeing with this positive correlation. So what we think we can do actually is pick apart the slope versus the intercept to start to see how much of a contribution the hippocampus is making versus a more non-hippocampal contribution. And critically, we can look at that sort of independently. So what we can use is general linear models to really assess the intercept versus the slope for these two correlations. So firstly, let's just focus on the slope here. So this is showing you the slope of that correlation for the no delay and the delay condition. Yes, there's a numerical decrease, but it's not significantly different, and we see a significant slope, positive slope for both conditions. But critically, if we look at the intercept, there isn't evidence for a positive intercept in the no delay. There is for the delay condition, and they significantly differ. So what this suggests, actually, is it's not that there's, as the non-hippocampal contribution is sort of going up, the hippocampal contribution is going down at the same rate. This seems to be additive in nature, in the sense that like what's happening is we've got the same slope, but it's just shifted up. So we've got an increase in this non-hippocampal contribution, but no obvious decrease in the hippocampal contribution. So it suggests an additive relationship where there are two contributions that are adding up to an overall reinstatement effect. And critically, it suggests that the time courses of these things might be different, okay? So the time course of the upregulation of a non-hippocampal contribution might be independent of the time course of the downregulation of the hippocampal contribution. So I'll get back to that in a second. Okay, so episodic events are still retrieved and reinstated holistically even after 24 hours. So we see that non-target reinstatement following a 24-hour delay. The hippocampus still contributes to that neocortical reinstatement because we still see that positive slope. But critically, there's this sort of additional non-hippocampal contribution which is seen in that positive intercept. So I think what this means is the hippocampus and the neocortex, or some non-hippocampal contribution, are both contributing in an additive manner to reinstatement following a 24-hour delay. And I think ultimately what's probably more likely is that there are independent mechanisms for the upregulation of the non-hippocampal contribution and the downregulation of the hippocampal one. So the upregulation of a non-hippocampal contribution is probably driven by, for example, sleep-dependent systems consolidation processes that are thought to occur, whereas the downregulation of that hippocampal contribution might be completely mechanistically independent. It might just be driven by decay mechanisms and forgetting mechanisms in the hippocampus. So they might not be tightly connected to each other. So what do I think this means? Well, ultimately, I think at the moment we have this sort of naive assumption that we start off in the hippocampus and we move in this sort of linear fashion towards a non-hippocampal source as a function of systems consolidation. But what I'm saying is actually that there are two independent contributions to that episodic reinstatement or that neocortical reinstatement. One is driven by the hippocampus, which can be weak to strong, and one is driven by non-hippocampal contribution from weak to strong. And by and large, we probably start off in this top left-hand corner and we move to this bottom right-hand corner. And I think often we assume that there's a sort of straight line or vector from one to the other. At the very least, what I think is likely to be the case is that this is non-linear in nature, in the fact that the trajectories, the time courses for the increase and the decrease will differ because they're different mechanistically. But I think once you start conceiving it of this two-dimensional space, I don't see there's any reason why we can't have episodic memories that start in different locations within the space and move in different directions if we appropriately kind of tweak experimental parameters. So that's kind of where I'm at now, and trying to think of ways in which we could kind of fully explore this space rather than focusing on this kind of unidirectional movement or unidimensional movement from this top left-hand corner to this bottom right-hand corner. So thank you for listening.
That's great, Aidan. Thanks very much. Any questions?
It was really wonderful. My question is, did you repeat this experiment after a week to see whether the slope
No,
spread further?
that's what I need grant funding for. Yeah, I mean, if what I'm saying is right and there are different sort of temporal trajectories for this increase and decrease, then realistically we need to do this at like multiple delays and actually track track the curvature of that increase versus decrease, and potentially see if we can modulate that time course with different sort of experimental manipulations. So yeah, there's a lot of work to do that I need money for.
Because what I found interesting was, even though statistically the slopes were not different, the correlation did drop compared to the previous thing. So I was wondering whether that's related to forgetfulness or whether neocortex is taking over the hippocampal thing. What is your...
Yeah, I mean, ultimately, I think it's additive in nature, but like if the standard time, like this is probably the standard trajectory that occurs. Like, you know, if everything's left to its own devices, I think system consolidation is probably largely correct, but with a slight, you know, a slight bow shape rather than narrow shape. That wouldn't necessarily be particularly controversial to say. But ultimately, I think we can start to conceive of ways in which we might be able to manipulate sort of the start and end point of this to try to really, like, I don't know, maintain the hippocampal contribution whilst a non-hippocampal source is upregulated, or bring back the hippocampus in certain situations to upregulate the hippocampus once it's been diminished and stuff. So I'm trying to kind of think of interesting ways to mess about with sort of the start and end locations and see how much flexibility there is within this space, basically. Like, is it always the case that it moves in this direction, but it's just we can mess about with the straight versus bowed line, or can we actually kind of start over here and move in this direction? You know, I think this is an unexplored space that we need appropriate experimental manipulations to explore
Very
properly.
interesting. Thank you.
Thanks, Aidan. Very clever stuff as always. Just wondering though, you are assuming that system consolidation is true, that there is downregulation of the hippocampal involvement, and there are some theories like multiple trace theory from Adele and Moscovich that assume the hippocampus plays a role continuously.
Yes.
So yeah, the question of a longer delay, saying with actually getting evidence for downregulation would be important, but also re-encoding. So if you have another retrieval test and then an immediate one, then a delayed one, or one after several days, which might involve a new trace informed hippocampus, would you expect a different
you're
result?
writing the grant for me. Yeah, so I think there's two things to say. One is that I want to explore the space, but two, I think you can basically use this space to think about any current systems consolidation model. So multiple trace theory would say we would always be here. Trace transformation theory would probably suggest that we should move in this direction, but actually, ultimately, I think there's a third dimension here of perceptual specificity of reinstatement that I haven't talked about today. You're right, like we might end up in this situation if you don't retrieve a trace for a long time, but that mere act of retrieval might suddenly upregulate the hippocampal one. So I think this offers a kind of, rather than a kind of mechanistic account of what might be going on, actually this offers a kind of framework for thinking about how all of these systems consolidation accounts kind of overlap with each other, with the similarities and the differences. So yeah, hopefully that answers your question.
Any other questions?
Hey Adrian, thanks for a great talk. I wanted to ask about the hippocampal deactivation. Do you think that's going to be more prominent in the posterior or the anterior hippocampus?
I don't know is the upshot. We haven't done systematic investigation. I mean, the original study, the sort of sweet spot in the hippocampus, I mean, so I did 2 analyses to show that original correlation. One was just taking the hippocampus as sort of ROI. One was just doing a whole brain analysis, and you basically only see that the hippocampus correlates with this neocortical reinstatement. I mean, little bits here and there, but the hippocampus is the biggest one. Of that whole brain analysis, it was like quite mid-hippocampus, so not obviously posterior versus anterior. Obviously, there are, I mean, coming back to Rick's question as well, there are other suggestions that it might sort of move from posterior to anterior as things get sort of more generalized over time, and that's something else we can look at, but we haven't looked at it systematically yet.
Great. Thanks very much, Aidan.
Thank you.
Right, so it's a pleasure to introduce the next speaker, which is Thomas Walbus from the German Center for Neurodegenerative Disease.
Can you say it in German, please?
No, I got it right.
All right. Well, thank you very much, Dorothy and Alex, for the kind invitation. So first time at BNA, my first time in Liverpool, and enjoyed both of them. And yeah, we're going to switch gears now a little bit, and then we're going to talk about our work in the context of aging and preclinical dementia. So when you look at the field of cognitive aging, there's obviously it's a huge field, it's huge literature, and people have done very beautiful and meticulous work on trying to characterize trajectories of how various cognitive abilities decline across the lifespan. And oh yeah, this pointer here. And I'm not going to go into the details here, but one thing that is quite striking is that when you look at things that we are interested in my lab, like spatial navigation, for example, does comparatively little work. Yeah? So when you find plots like these where I talk about spatial orientation, typically what's measured is like spatial functions, simple 2D tasks like mental rotation, for example, which is very different from moving around and keeping track of where you are. However, it was already Charles Darwin who in 1873 speculated or actually observed that the sense of direction is sometimes severely disarranged in older and feeble persons, and he used that observation to speculate that there might be something like a specialized part of the brain that's specialized for the function of direction. So when you look at the emerging literature on navigational decline, one thing that becomes very obvious is that that the variability in people's performance increases as people get older. So this is data here from a virtual Morris water maze task where people or subjects would be navigating a Morris water maze in VR, just on a screen in this example, and you have like age on the x-axis here and the total distance traveled to find the platform on the y-axis across trials. And what you see is obviously that as people get older, the performance variability seems to increase. Yeah? So some older adults who are severely impaired who take a lot of time or travel a lot of long distances, whereas others are at levels of performance that's actually quite similar to people in their 30s and 40s. We've seen our data actually very similar, I think maybe more striking. So this is a study that we published a couple of years ago. It was actually from an imaging study where we had subjects learn the inner city layout of Tübingen in southern Germany. So this is a virtual model of Tübingen. You see a bird's eye perspective here. And what happened is that after experiencing this environment from a first-person perspective several times, they would be positioned somewhere in the environment and they'd be asked to now point to an unseen location. Yeah? As if I would now be standing here and had to point to, I don't know, Royal Liver building outside, right? And what you see here is across learning blocks performance, basically something like angular pointing error, yeah, across these sessions. And in gray you see young adults, and in orange you see older adults. And you see that, yeah, there's some that young adults very quickly learn, and then they reach some kind of floor effect. So basically they're able to point within, say, 10, 20 degrees of error typically. Whereas in older adults, we see a large variability. Some older adults actually do reach similar performance levels, but others really stay at very high levels, so they produce very large errors throughout, in this case, 8 sessions. Yeah. So that's a very typical finding. And I would like to explore today with you what could be just one potential mechanisms that could contribute to this variability here. And in order to do so, we're going to focus on grid cells in entorhinal cortex. And This was the first electrophysiological evidence for grid cells in humans, provided by Josh Jacobs at Columbia, who basically recorded in epileptic patients while they were doing a virtual navigation task and found or observed firing patterns in entorhinal cortex that were reminiscent of grid cell-like firing, as had been obviously described in many, many beautiful experiments in rodents. Since then, however, since this initial demonstration of spatial or pure spatial signal, obviously many theories have been advanced to think about, okay, how might these signals, these entorhinal grid cell signals, also contribute not only to navigation, but also in the service of episodic memory. Yeah. So this is a model put forward by Edward Moser and a colleague, where they basically said, okay, well, maybe what grid cells in MEC do is that they provide kind of positional tags that would be combined with, for example, temporal and item information from LEC to then give rise to like these integrated memory traces at the hippocampus. There are other models, like from Neil Burgess and Andrew Pikansky, where they talked about recognition memory and that MEC cells could provide something like 2D Saccard vectors. There's obviously also work like geometric coding, and more recently there was a model by David Huber where even diverge completely from spatial information and say actually the key information that you see in grid cells is what information that is actually at locations where a non-spatial attribute is remembered. So we're interested to think about, okay, what is the role that potentially altered grid cell computations might play if we observe them in cognitive aging. So this is the first study we did on that a couple of years ago. We first looked, okay, what does this grid cell-like signal look like in humans? And for those of you who might not be familiar with the idea, so the idea is basically that when you do fMRI experiments, which is what I'm presenting here, obviously we don't have access to like single cell data, but there are some underlying assumptions that make the claim that what you should observe is some kind of periodically fluctuating signal as a function of running direction in the environment. Yeah? So when you move through, say, some virtual environment, you should observe a bold signal that basically goes up and down in multiples of 60 degrees. And that's due to the organization of the grid fields. I'm happy to elaborate on this more, but that's the general idea. So you can basically have subjects navigate, in this case in a virtual environment. You track their movement direction as they move about, and then you look for a signal that might show stronger or larger amplitudes in steps of 60 degrees, right? So this is what Neil Burgess and Christian Döller had published in, I think, 2010 it was, the first time, and there's been many replications since. And we basically did this experiment here, and we looked at younger adults, and we could replicate the effects that we also see this hexadirection modulation, whereas in older adults at the group level, it was basically absent. Yeah, so we didn't see any kind of significant hexadirection modulation there. We also saw some correlation with performance. We don't have to go into there. But what was really interesting is that actually when you split these older adults by their navigation performance, so we did just a simple median split there, and then we actually found that, oh, wait a moment, there's this subgroup of older adults here who have very good navigation abilities, that in this group we actually see a hexadirectional signal whose amplitude is quite comparable to what we see in those younger adults. Yeah? So it seems that these older adults, again, are not a homogeneous group, but there's quite some variability, and some people actually are able to perform at similar levels. Okay, so this got us very interested in a new phenomenon, not a new phenomenon, but a recent phenomenon that's now being studied in Magdeburg quite intensely, and that's the phenomenon of superaging. In case you haven't heard about superaging, so there's a, was probably coined at Northwestern, where they basically describe people who are aged 80 or older, and whose episodic memory performance is at the level of 50 to 60-year-olds. Yeah, there's a whole literature now on superaging, and it's isolated to episodic memory. So basically, when you look at other cognitive domains, say working memory, attention, perception, they perform at comparable levels to their age-matched peers. It's just this kind of isolated episodic memory effect where they seem to outperform. There have been a number of studies to try to explain what could be potential cortical or subcortical mechanisms that could give rise to these effects. And people have found like larger brain gray matter volumes like an anterior cingulate in the hippocampus, more von Economo neurons in ACC, there's some DTI work. And for the context of today's talk, I just want to point you to this study here, which is a post-mortem study where they actually did staining in post-mortem brains of superagers and typical agers. And what they report in that study is actually that both neuronal size in entorhinal cortex was actually larger in those superagers, plus that they showed less tau pathology. As you can see here, so lower tau pathology in the EC, and also neuronal size being larger in the brains of these deceased superagers. So in Magdeburg, we've now built up a huge cohort, a molecular characterized human aging cohort, which is kind of a large collaborative project that involves by now about 500 participants. We're still including subjects in that study, and it's quite a comprehensive assessment. And I just want to show you some grid cell data that we've recently gotten from a subgroup of these participants here. So we're now doing a similar task, except that we now do a passive navigation task, because a critical issue with these aging navigation tasks in VR and in scanner is that older adults, when you give them freedom to move, if they have a joystick and they can move around freely, you get quite a lot of variability in behavior. And there might be some people who are very hesitant, they move a little bit, they stop, they wait for 5 seconds, they move again. So it's very tricky to exert tight experimental control. And therefore, what we did here is we moved to actually passive navigation task, where they're basically observing kind of a video. Yeah? So they're seeing movements through this environment. And their task is to memorize the locations of objects that they encounter here. Yeah? So you see this desk or this bench here. So there would be several objects scattered throughout the room, and they're instructed to remember the locations of each of those objects, because they will be probed on their memory later on. This has the added advantage that we can precisely now control the trajectories that people are experiencing, and we can precisely match that, for example, they're experiencing each of those movement directions at the same frequency, yeah? Which again is difficult to do when you give them free. So these are baseline controls. These are people aged 60 to 70 here. We're again measuring now the magnitude of this hexadirectional signal. And we're seeing kind of similar to what we've seen before. And in superagers, we see an effect that's quite similar. So these are, again, now people 80, some of them 90, early 90s. Yeah? And we see a hexadirectional effect that's quite similar. Whereas those typical agers, so these are now age-matched controls that do not fulfill those criteria of being superagers. So they show normal episodic memory performance. And we see a much lower signal. After scanning, we then give them 2 assessments. So we try to probe their knowledge both of spatial and of item information of the objects they have learned. So this is the first what assessment. It's kind of similar to this classical pattern separation task that Mike Yasser and others have developed, where we show them objects they have encountered, in this case the bed, and also slightly altered versions. So lures, stimuli, so stimuli that have been altered. And they basically have to make a judgment for each object whether this is the exact stimulus that they had encountered in that virtual environment, or whether it's an altered version, it's a lure, it's not a stimulus they had seen before. Yeah? So similar to Mike Yasser's pattern separation task. And we can then basically compute D primes as kind of a measure of sensitivity, how good people are at distinguishing signal from noise. And we basically see that those superagers are on par with the baseline controls and definitely outperform the typical agers, and that across groups, the strength of this hexadirectional modulation seems to be related to their sensitivity to be able to remember this item information. In contrast, which is interesting, when we then look at wear memory, in this case, the way we did it is that we now showed them an overhead view of the environment. We showed them the object, and they now had to place it. Yeah? So they basically would tap on a touch screen at the location where they thought this object had been presented. It's a little bit hard to see, but you see the landmarks on the walls here that allowed subjects to orient themselves. And there we find no evidence for difference between those superagers and those typical agers. So it was, at least in this data set, restricted to this water information. Okay, now we want to take these insights into entorhinal dynamics and entorhinal changes in aging and move to the direction of preclinical Alzheimer's disease. I guess you've all heard multiple talks here and probably at other conferences where people talk about, well, when we think about Alzheimer's disease, when we look at progression of disease, various disease indicators, we have this problem that, okay, there are certain indicators like, say, CSF-based amyloid, CSF-based tau, or what you can measure with PET. We see early changes, But there's a long stage where cognition, for example, as shown in green here, this green here stays relatively normal. Yeah. So can we, there's a long discussion in the field, can we somehow develop cognitive readouts, cognitive measures that might be more sensitive to those early disease stages? So there has been recently very beautiful work, maybe Coco will also talk about that, on like early MCI and studies by Dennis Chen, for example, who showed that there are ways to improve on that. We were interested here in this phase, this pre-symptomatic phase, in this phase where at this stage at least there are no cognitive deficits observable, at least with standard neuropsych tests. Yeah. So we turn to a phenomenon that's called subjective cognitive decline. So people who subjectively report that they feel their memory is fading, their memory is getting worse, but they show no impairments on standard neuropsychological tests as they would be administered in clinical context at the moment. Yet there is some evidence, for example, some work on tau PET, showing that at least some of them actually do start to show enhanced tau pathology in entorhinal cortex. So there might be things, 5 minutes, okay, going on that are not picked up yet. So we are interested in that. So what we do, very quickly, we turn to a behavior. It's called path integration. In case you're not familiar with that, path integration is a very simple behavioral readout where you have to keep track of your position by integrating self-motion cues. So a typical task would be you're being led along some circuitous trajectory, and at the end of the trajectory, you have to tell us where you started from. You have to maybe go back, make a beeline. You can do this blindfolded. We did this here in immersive VR, where this would be a subject. There would be this very featureless environment, and this is kind of a floating ball. And what you have to do is you have to follow the ball. And the ball basically floats on some circuitous trajectory, and then at the end of the path, you have to point back to where you think you came from. Very simple behavioral assessment. And we tested about 100, or Vlada Segan in my lab tested about 100 participants in that study. And as expected, in all these kind of standard neuropsych tests that we did, there was no difference whatsoever between those SCD patients and our healthy controls. We then did 2 path integration assessments. First one based on previous work, looking at angular encoding. So how good are people at keeping track of their orientation as they move along this path? We stopped them twice. And in this study, we do not see any differences between these SCD patients and healthy controls. They were both able at a similar level to basically do these angular judgments. And this is in agreement, at least with some rodent findings, say mouse models of tau and amyloid pathology, who also showed that head direction cells that obviously are assumed to be critical for orientation coding seem to be relatively unimpaired in these mouse models. However, when we looked at the overall path integration response, where they now have to tell us where exactly was your starting location, now we are starting to see differences. Now we're starting to see that this SCD group actually shows much higher path integration errors, and that this was also correlated with neurofilament light chain as we deduced from blood sampling. So that's interesting. But even though path integration is a very simple behavior, we wanted to dig a little bit deeper and try to understand, okay, what is it exactly? What is kind of the computational mechanism that could give rise to those path integration deficits in those SCD patients? So we teamed up with Zoran Tiganj from Bloomington University, and we basically developed an extension of a computational model whose aim is to decompose path integration error into things like movement perception, into things like response computation, into things like accumulating noise, all little sub-processes that can compromise your performance in the end. And what we're finding is, maybe a little bit hard to see, but there was a selective difference only in one parameter in this model, which is memory leak, which basically suggests that there might be a leakage in the integrator as you walk along this path, that your integrator, which kind of represents past states, is leaking, and that this contributes the most to the errors that we're seeing in those STD patients. All right, so I think we need to come to an end now. So we do assume at this stage that maybe preserved entorhinal computations could be just one key mechanisms contributing to superior episodic memory in superages. Of course, we need to follow this up with a larger sample, but I think the data are interesting at this stage. At this stage, however, we have no evidence that superages are also superior navigators, which is very interesting, because you might think, well, if grid cells are responsible both for its episodic memory and navigation components, and they should show both. At this stage, we have not seen this, which is quite interesting. But of course, you could then develop strategies to maintain enteral integrity if, for example, something like preserving cognition in old age is of interest to you. The second part, I think, is now nicely converging across labs and species, because there's also beautiful rodent work suggesting that path integration could be a very sensitive marker for early disease stages in the context of Alzheimer's disease. And deficits in path integration have also been found in younger adults with increased genetic risk of AD, and Coco might allude to that in this talk. And so the idea could now be that maybe changes, if you were able to measure changes in path integration in a clinical setting in a very controlled environment, could provide sensitive indicators of early disease pathology, slash also, if you're interested in tracking disease progression. Yeah, and I think my time is up. Thank you very much for your attention, and look forward to questions.
Thank you so much, Thomas. Any questions?
It was really interesting. My question is, what are these super agers doing differently compared to normal agers?
So it's probably a multitude of factors. There's not a single factor that can explain that. And I think at this stage also the evidence is not yet strong enough that we can conclusively say it's this particular. Brian Strange, we might know from Madrid, they published a quite nice study last year where they actually looked at a whole range of factors. And one of the factors that they isolated was actually mental health. So things like depression and anxiety had a very strong predictive value. Social engagement as well, and then of course your usual suspects like physical activity, nutritional status, these kind of things. But I highly recommend looking up this paper. I should also say what I've provided you now with is one definition of superaging, which is quite common, but it's not, there's no uniform agreement at this stage how exactly to define superaging. And when you look at the literature, you'll see different criteria being applied across studies. So I think at this stage it's a little bit too early to really make this.
Thank you.
I think Rick and you.
Yeah, lovely work. So the question's quite specific, but I'll explain why. So for the fMRI grid cell analysis, you need to split half your data and work out the orientation one, apply it to the other. Yeah. I was just wondering how you did that. Like, is it first versus second half of the experiment, or is it sort of interleaved?
Right.
So the reason I'm asking is because there's two reasons why you might not see a grid-like signal. One is that there isn't one. The other is that it actually drifts over time.
Exactly.
And if it's the latter, I wonder whether that might be an explanation for like the sort of memory leakage phenomenon that you're seeing behaviorally, where they do have a grid representation, but because it's drifting, you can't really like align it with what went previously, so you're getting this kind of memory leakage.
Exactly. So, in this superager work, this is a paradigm that's relatively short, so it involves two independent runs. And basically what we do is going to be training on the first run, it's kind of stopped, and then testing on the second one. And then exactly what you said could happen, that if you have, say, a drift in grid orientation, and you then compute your grid magnitude, you will see basically no effect, right? Because the prediction doesn't match what you would expect. So this is what we saw in the first study, also in this first aging study, and this is also what seems to be going on in those agers, those typical agers that have much lower grid magnitudes there. Whether this could also explain this kind of drift or leakage in those path integration tasks, I think it's a little bit hard to answer this question, because don't forget, like, path integration is kind of a working memory task. It's a very short term. It takes you like maybe 20 seconds to walk along this path, and then you have to go back. Whereas in those grid cell tasks, these are kind of tasks that span like multiple minutes, like 10 minutes would be one run. So where the temporal dynamics of a drift and grid is something you can't really capture with this entire evidence.
Rick.
Is it time?
Yeah.
Great stuff. Thank you very much. So just going back to superagers, and it's a comment and then a question. And the comment about definition of superagers, I know you understand, but I say it so I can justify my question. The schematic you showed with superagers being flat and typical aging declining, of course, the standard definition of superagers doesn't discount the possibility that the superagers always started higher.
Sure.
So there's no difference in decline, you just have a higher baseline.
Yeah.
So the question is, in your new study, are you able to track these people over several years, look at longitudinal changes? That's a much better definition, I think, of super ages.
Totally, fully agree with you. And indeed, so this cohort study that I introduced, so the hope is, if the funding comes through, that we'll be able to follow them for 12 years. We are actually now re-inviting those super ages. We tested these data. The first ones were tested 2.5 years ago. We're just now re-inviting them to start to track change. But hopefully we get more than 2 time points, 3 or 4 in the end. And yeah, that's the idea.
Great. Thank you so much, Thomas.
Thank you.
It's my pleasure to introduce our next speaker, Coco Newton from the University of Cambridge and also UCL.
Thank you very much, Dorothy, and for organising this symposium. So I'm actually going to pick up basically exactly where Thomas left off, so thank you very much for doing my introduction for me. My talk is split into sort of 3 sections. So first, fleshing out a little bit what Thomas mentioned earlier about why theoretically navigation could be relevant in Alzheimer's disease and how animal and human studies provide a bridge. Then some actual evidence for there indeed being a role of navigation in Alzheimer's disease, and then how do we take that forward? How do we translate this into different types of impact in science and beyond? So for the first bit, a little bit of a primer on Alzheimer's disease. I'm sure you all know it, but on the left-hand side you have a healthy brain, quote unquote, and on the right-hand side is a brain that has had several years of Alzheimer's disease pathology causing atrophy and cell loss. It's really pressing at the moment because we do have some treatments that purportedly could slow down disease, but current estimates suggest that only about 2% of people would be able to access it if it were made on the NHS, made available on the NHS tomorrow, and that's kind of a function of problems in diagnosis and detection. On the other hand, 50% of cases might be preventable, so there's an argument that we should be detecting people at high risk. And it's also the leading cause of death in the UK, and it's slowly becoming the same across the globe. There's different models of the life course of the disease, and Thomas showed a more detailed one showing how the different biomarkers go off over time in the run-up to dementia becoming apparent. But if we take a more simplified version, you can imagine that if time is on the x-axis, there are sort of 3 main groups of people in life. People that will progress and never accumulate any biomarker evidence of Alzheimer's disease, so no misfolded proteins. People that will start to accumulate misfolded proteins in the brain, but they will never really reach a clinical threshold for dementia. And then there are some people that at some point will start to progress up the dotted line, and that's a very dynamic process. We don't fully understand it yet. There's lots of reasons why you might progress up. For example, your genetics, cognitive and brain reserve, and there are lots of factors indicating that you might be progressing up it, and that could be imaging changes or it could be cognitive changes. But the real problem is that the current detection point is at quite a late stage along that trajectory, almost at the point that you're basically being diagnosed with dementia. But theoretically, if we wanted to have a chance at those new treatments being able to slow down the disease in people and prevent your brain becoming the atrophied one on the left versus the healthy one on the right, we should be detecting people at this much earlier inflection point here. So how does navigation and animal work have a role? What could we do with this? So if we take a very theoretical approach and we say we know Alzheimer's disease is progressive, where does it start? And there's a lot of evidence suggesting that one of the misfolded proteins, tau, begins in the entorhinal hippocampal circuit. And that is also a region of the brain where we have these grid cells that Thomas mentioned. And they were discovered from recording in the rodent brain with single-cell electrodes, overlapping it with the position data of rats as they were foraging in a box. And then you get these nice heat maps showing a regular firing field pattern that is akin to a grid. And the theory goes is that that provides you with some sort of coordinate system by which you could potentially measure vectors in space, so tell you how far and in what direction you've been traveling. And then later studies, after the discovery of these cells, did the same experiments, but this time in rats that had been transfected with the human tau gene. And then they found that this grid completely destabilized. And it's almost as if there was a lack of that coordinate system. And in similar studies, they looked behaviorally, how does that impact the rodent's ability to perform a navigation task in something like the Barnes maze, but it's been repeated in a lot of different paradigms. we find that the model rats or the model mice completely lose the ability to do goal-directed navigation. So, if this is what is happening in early stage tau pathology in rodents, then might we see the same observation in humans? And moreover, it's not just the entorhinal cortex and grid cells that might be relevant in Alzheimer's disease. There's almost a sort of 100% overlap between different regions of the brain that confer spatial abilities and the progression of tau pathology and amyloid pathology in the brain, as outlined in this really nice review by Gillian Coughlin. So there's a wealth of evidence to suggest that navigation should go off in Alzheimer's disease. So what is that evidence? We designed a path integration task in collaboration with Neil Burgess's lab. And so this is similar to Thomas's task, but not identical. This is the classical triangle completion task, where participants in immersive virtual reality are guided between 3 cones. And then when they hit cone 3, they have to try to get back to cone number 1. And in reality, participants might take the orange line instead of the white line, and then we can measure that error between where they thought they were and where cone number 1 actually was, just in a simple location error. And then the other thing about this task is, because that's a relatively straightforward task, we wanted to sort of stress test the entorhinal cortex in these individuals. So we had different conditions for the task. In the baseline task, the environment stayed the same when they got to cone 3 and they had to go back to cone number 1. In the second condition, we took away the floor texture. So as people are moving, there's no optic flow. It makes it really hard to estimate how fast you're traveling, which is one component of using your self-motion information. And then in the third condition, we took away the orientation cues from the horizon, which makes it very hard to estimate which direction you're facing. Again, the other component of self-motion information you need in path integration. And so in the lab before me, there was a student, David Howitt, who took this test and gave it to a cohort of individuals that were either healthy controls, average age of around 70, and then individuals that had some degree of cognitive impairment. And in those people with cognitive impairment, they either had biomarkers for early Alzheimer's disease or they did not have biomarkers for early Alzheimer's disease. And what we saw is that the individuals with biomarkers who had cognitive impairment, the ones in red in the middle, had a really increased error on this task. So their location error was much worse than the other 2 groups. And then when compared on a battery of different cognitive tests, this test was much better at differentiating those 2 groups of individuals. So that was a nice sort of piece of evidence that suggested, indeed, maybe path integration is having a role in Alzheimer's disease, and it might be a sensitive indicator. But that's not the most interesting question. The most interesting question is this the inflection point right at the start when people on the dotted line start to diverge from other people that have biomarkers but won't progress to dementia? So in my study, we repeated this, but then we took people about 20 years younger than that population, and then we stratified them by 3 sort of main risk factors that you can measure in midlife, whether or not they have an ApoE4 allele, which is about a sort of 15% increased risk of future Alzheimer's disease, family history of dementia, and the CADE dementia risk score, which is a multifactorial lifestyle-based risk score for future dementia that I'll explain in a minute. So we didn't actually find that individuals performed worse on this task in each individual condition. So what we wanted to do is look at, if you start to take away cues from the environment, might that uncover subtle changes in path integration performance? So we looked at the change in performance from the baseline condition to the condition either where we took away the floor texture information or we took away the orientation cues. And what we found is that when we took away the orientation cues, that's when we started to unmask a much larger location error in the individuals that have both a family history and an ApoE4 allele, so i.e. the highest risk group that we could create in this midlife cohort of 100 people. And then we also included a sex interaction in our analysis because we know that there's a huge difference in risk for future dementia based on your sex. And we found that this was an effect specific to the males in that high-risk group. Then, as Thomas said, there's different components of why you might begin to make errors in a path integration task. So we decomposed it into, were people turning, were they underturning or overturning, or were they overwalking or underwalking to get back to cone number 1? And in contrast to Thomas's data, we actually found it was more of an angular error, so people weren't turning as precisely in order to make it back to cone number 1. We can discuss that afterwards as to why we think that might be. And then finally, it's not just an effect in the family history and the ApoE4 risk group, it was also an effect in the lifestyle group. So on the left here, you can see what goes into making up someone's CADE score, and if you have a higher score, you have a higher lifestyle risk. And we found that the higher your lifestyle risk correlated with having a higher location error when we take away the orientation cues. And we didn't find any effects for risk factors when we took away the optic flow, suggesting it was something about orientation information in the environment and your ability to get back to cone number 1. We also gave these individuals some other navigation tests because, as I said earlier, it's not just entorhinal cortex that might be implicated in the relationship between navigation and Alzheimer's disease. There are also other navigation functions like allocentric memory, so remembering information about your environment regardless of what viewpoint you have, or indeed having a memory of the environment based on what your viewpoint is. And we found that individuals with family history performed worse at an allocentric spatial memory test. And we found that females with a family history performed worse at an egocentric spatial memory task. And there might be something to do with the distribution of tau pathology or the rate of tau pathology accumulation across genders or sexes that might be explaining that effect, but we need to confirm that in some future work. The interesting thing, though, is when we gave these individuals other tests, so the Addenbrook's cognitive exam for global cognition, narrative recall, or a shape color binding associative memory task, we didn't find any of the same risk effects that we found in the navigation tasks, specifically path integration. And it's not just these VR navigation tasks that have these effects. We did a systematic review where we looked at every single study that had reported some sort of group difference in either individuals that were asymptomatic but had risk factors for future Alzheimer's disease, or they were symptomatic and they had future risk factors for Alzheimer's disease. And we found a whole host of studies that seemed to have some sort of sensitive differentiation of these different groups. And nicely, it's not just immersive virtual reality that these tasks are done in where they find effects. It's a whole host of different tools suggesting that regardless of how you do these tests or in what format you do these tests, you are likely to find some sort of effect. So what do we do with this information, and how does that have implications for the bridge of animal and human research? Well, I think if path integration changes are that sort of initial inflection point in the disease course, that potentially there's something about entorhinal computations that is relevant in a very early stage of Alzheimer's disease, then we need to understand why that is before we can translate that into a meaningful clinical tool. And so that's where I think translating backwards into animal studies could be really nice, because in essence, a navigation paradigm in VR that you can do in humans, but you can also do it in mice, it provides a platform by which you can give the same paradigm, you can have the same outcome metrics, and in humans you can infer what's a clinically relevant behavioral change that you need to measure. And then in the rodents, you can begin to understand the cellular underpinnings or the network underpinnings of that behavioral change. And then even more sort of relevant for not just diagnostics, but treatments, then suddenly you have a platform by which if a drug starts to have an effect at the cellular level as well as the clinical level, that's a much better indicator that that drug is doing something meaningful than giving someone a drug and asking them to remember names and addresses. The second direction that you can go is understanding mechanistically what it is that's going off in the brain in relation to Alzheimer's disease and navigation. You have to think about scalability. So it's not reasonable that we're going to put an immersive virtual reality system into every GP surgery, but we might be able to get close to that with new technologies such as augmented reality. So Andrea Castagnaro, who is in Neil Burgess's lab, is working on some modeling work to understand what it exactly is the angular component of path integration that might be going off. Is it even angular component of path integration? And we're looking at these new augmented reality apps, a bit like Pokemon Go, where you can start to sort of just do rotational on-the-spot tasks, which might derive the same outcome measure and might be probing the same computations. At the same time, if we want to be moving into the clinic, it's not just going to be neuroscientists that's going to get there, it's going to have to be a collaborative effort. So at the moment, we're working on a study where we've incorporated experts from a variety of different backgrounds, people that work in patient safety, but also people that work in systems and health systems design, who are, on the one hand, validating the science, on the other, making sure that the device that has the test is as usable as possible for doctors and for patients, and then also thinking about the service design. So how are we going to implement a digital technology into a GP surgery? What are the constraints? What are the factors that we need to be thinking about here? And specifically, we're looking at that in relation to this task at the bottom, which is called the 4 Mountains task. And then the third way I think that we can translate based on this understanding is going from what we observe in the clinic and in the real world and what people tell us are their problems and going back into science to design studies that are a little bit more real world relevant. So we've been doing a lot of interviews with people with lived experience of dementia to sort of map out the entire journey pathway of going through a diagnosis of dementia. And so far I've been telling you about this observation that clinicians might not have sensitive tests in order to detect those very early symptoms. And that is where navigation could have an impact. But there's actually several other places where navigation could have impact in the context of Alzheimer's disease. And that's in the very, very early stage of the journey when people are noticing symptoms. A huge problem is people don't know what a symptom of dementia versus normal aging is. So could we be thinking about navigation tools like an AR game that people could just access at home and just rule out the fact that, no, what you're experiencing, it's not related to early stage Alzheimer's, it's something else. And could we even look at passive ways of collecting people's GPS data in an anonymous way to derive sort of patterns in their everyday location behavior that's potentially relevant in the same way the cognitive test could be. And then the third, on the opposite end of the spectrum, if you start to lose your navigation ability, it's incredibly dangerous, and it's also a huge reason why you lose independence as you get older. So this is a study from Michael Hornberger's lab where they looked at the reports of people getting lost in Norfolk and then matched it to the street network entropy of the local region. And they were able to sort of map different parameters of the environment to the incidence of people getting lost. They worked together with the police force in Norfolk. And I think this is a really innovative way that we can think about using navigation neuroscience for a much more real-world impact than perhaps we have previously considered. So just to sum up, I think that the human-animal relationship that the navigation enables us to sort of probe is really important for this mechanistic understanding of Alzheimer's disease. We know now that we have these repeated observations that navigation does go off in some form in Alzheimer's disease with different tools, different tasks. And I think we can afford to think a little bit more broadly about how we can translate navigation neuroscience, not just from animal studies into humans, but from humans back into animals, from humans to the clinic, and from the clinic back to humans and animals. So I'll stop there and happy to take any questions.
Thanks, Coco. That's great. Right, lots of questions.
Hi, thank you. My question is, can we do these tests on a computer rather than using virtual or augmented reality? Because I'm thinking if the patient is old, the mobility is also one of the issues. So we may not know whether they're not able to do that because they have arthritis or some other things going on. What are your thoughts on that?
Yeah, I mean, so in the systematic review, there were multiple studies that used desktop computers to do these tasks. It's also a question of what's the right test for the right target user. If you're talking about people that have diminished mobility, then perhaps a cognitive test of navigation isn't the right one to use.
Question for Coco and I think Thomas as well. To what extent do you take into account people's navigation skills or in real life? And the extent to which they navigate, do you log the number of miles they drive, or are they cyclists, or are they ramblers or hill walkers? Does that have a bearing on your findings at all?
I think it's something that we don't do enough in navigation research. It's also quite hard to measure. I mean, people are typically really bad at self-reporting their navigation ability. So yes and no. I mean, we know that there is a huge component of life course navigation experience that influences your ability. So there's data from See Her Request, which is a citizen science project from Hugo Spears and Michael Hornberger, where they essentially had a data set of a couple of million people, and they were able to show that the Nordic countries had slightly better performance overall across all countries, and that might be because they have orientation as part of their curriculum, but that's still a speculation. Beyond that, I think that's where navigation researchers are moving now, is into that more qualitative element of trying to understand what does it mean to be a navigator in the real world. Then there's also some literature about navigation usage, GPS usage for navigation and how that affects your ability to navigate. So it's coming.
Yeah, so I have a question regarding the perspective regarding, so I'm building robots, so maybe one of the few people doing that and trying to understand animal behavior for a while. And path integration is usually the worst thing of doing that, because path integration obviously accumulates error. Yeah, so basically at the moment there's a landmark there, for those who are old enough to remember Breitenberg vehicles. Yeah, so at the moment there's a landmark there, you would use that. Yeah, and so therefore if you've got these nice triangles even in the VR space, I would use that, because I can create an error signal on this and home in on it. And so I don't need a bird's view coordinate system, which is terribly difficult to create. So how do you make sure that you don't that the people don't use that? And then I'm baffled that nobody has been talking in these 4 sessions about this at all, because for me as a robotics person, I would directly use that.
Yeah, so the specific virtual reality environment that we use in our task, the cues are actually projected at infinity. So if you keep walking towards them, you'll never reach them, which means that you can't use them for distance estimation, only for pure orientation information. So they don't offer as much as a corrective mechanism as you would expect in the real world. And moreover, that's why we also had the 3 conditions, so we could compare the effect of removing those supportive landmarks. In general, we know that, yes, absolutely, people and animals are rubbish at path integration over time, and they get worse, and that is corrected by a landmark. If you take APOE4 carriers, which have a higher risk of Alzheimer's disease, they tend to adopt a behavioral strategy of sticking near to the walls of environments in these types of paradigms, which seems to be a corrective mechanism for dealing with the error accumulation over time. So we tend to design the paradigms that controls for this and accounts for this. But the reason that we test this behavior is theoretically because of the entorhinal computations that we think support it. But in the real world, yeah, of course, we don't actually navigate using path integration. We probably combine multiple cues and strategies over time. And I think one thing we could be moving towards, and as I said, with translating back from the real world into the design of studies, is to make them more real world, because it's not very ecologically valid to test people on one specific strategy.
Great. I think we're out of time, Rick, sorry. But Coco, thanks very much. That's great. So, Dorothy, you have to come up as well. So that's just leaving us to thank 4 speakers who are actually some of the best at keeping to time I think I've ever chaired, so thank you for that. And some great questions, but again, from my point of view, it just highlights why EBBS is supporting this symposium in terms of all the speakers putting behaviour at the heart of the neuroscience and the understanding of the neuroscience that's going on, and what added value that brings to it. Dorothy, have you got anything else you'd like to say? Other than also an enormous thank you to Dorothy, who didn't think that being local co-chair was enough to do, and so wanted to help organise a symposium as well. So thank you, Dorothy.